Okay, so the moth and yucca plant cannot complete their life cycle without each other. Okay, the moth deposits its eggs in the locule of the ovary, and the flower in turn gets pollinated by the moth. Right? The moth deposits its eggs in the locule of the ovary, and the flower in turn gets pollinated by the moth. The larva of the moth come out of the eggs as the seed starts developing. Okay, why don't you observe some flowers of the flowering plants, or like it's all sheet eucalyptus, banana, and all these plants like. Okay, many insects may consume pollen or the nectar without bringing about pollinations. Such floral visitors are referred to as nectar robbers or pollen. Right. So outbreeding devices. Now, what are the outbreeding devices? The majority of flowering plants produce hermaphrodite flowers. Right. Hermaphrodite flowers and pollen grains are likely to come in contact with the stigma of the same flower, and the pollen grains are likely to come in contact with the stigma of the. Okay, so the majority of flowering plants produce hermaphrodite flowers, and the pollen grains are likely to come in contact with the stigma of the same flower. So continued self pollination results in inbreeding depression. If there is no cross pollination and keep on continued pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self pollination, because we want to bring some variation, okay, for the evolution. So to encourage cross pollination, in some species pollen release and stigma receptivity are not synchronized. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released released before the stigma becomes receptive, or stigma becomes receptive much before the pollen release of pollen. So first, so this is the first method to prevent the self pollination, right? Either the pollen is released. Okay, now in some other species the anther and the stigma are placed at different positions, so that the pollen cannot come in contact with the stigma of the same flower. So the position of the pollen, so the position of the pollen and the stigma is different on the same flower, right? Both these devices prevent autogamy. Now autogamy, it is the pollination of the transfer of pollen grain from the same flower of the same plant. Okay, so both these devices prevent the autogamy. Now the third device to prevent inbreeding is self incompatibility. Now this is the genetic mechanism and prevents self pollen from the same flower or the other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Now another device to prevent self pollination is the production of unisexual flowers. Now if both the male and female flowers are present on the same plant, such plants castor and maize, these are monoecious. So castor and maize, these are the monoecious, right? Castor and maize, these are monoecious, and it prevents autogamy but not the geitonogamy. Okay, geitonogamy can be occurred because if both and female flowers are present on the same plant, right? On the same plant. So castor and coconut not on the same flower. So male and female flowers are present on the same plant, such as castor and such plants are castor and coconut, no, castor and maize, right? So castor and maize, so these are the monoecious plants. It prevents autogamy but not geitonogamy. In several species such as papaya, male and female flowers are present on different plants each plant is either male or female, that is dioici. In several species such as papaya, male and female flowers are present on different plants. So male and female flowers are present on different plants, right? That is each plant is either male or female, that is dioici. This condition prevents both autogamy and geitonogamy, right? Now pollen pistil interaction. So we are going to study the pollen pistil interaction, right? So the pollen pistil interaction. So pollination does not guarantee. So So the pollination does not guarantee the transfer of the right type of pollen, that is compatible pollen of the same species as the stigma. Often pollen of the wrong type, either from the other species or from the same plant, if it is the self incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is of the right type, compatible, or of the wrong type, that is incompatible. Right. Right, if it is of the right type, the pistil accepts the pollen and promotes the post pollination events that leads to the fertilization. If the pollen is of the wrong type, the pistil rejects the pollen by preventing the pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between the pollen grain and the pistil. Okay, this dialogue is mediated by the chemical components of the pollen interacting with those of the pistil. It is only in recent years that the botanists have been able to identify some of the pollen and pistil components and the interactions leading to the recognition followed by acceptance or rejection. As mentioned earlier, following compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pores. The contents of the pollen grain move into the pollen tube. Pollen tube grows through the tissues of the stigma and style and reaches the ovary. You would recall that in some plants pollen grains are shared at two cell stage, a vegetative cell and a generative cell. In such plants, generative cells divides and forms the two male gametes during the growth of the pollen tube in the stigmas. Okay, in plants which shed pollen in the three cell condition, pollen tubes carry the two male gametes from the beginning. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus. Many recent studies have shown that filiform apparatus present in the micropylar part of the synergids guides the entry of the pollen tube. All these events, from the pollen deposition on the stigma until pollen tubes enter the ovule, are together referred to as a pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. So, as pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. Right, the knowledge gained in this area would help the plant breeder in manipulating pollen pistil interaction even in incompatible pollinations to get the desirable hybrids. Okay, so it can be used in the incompatible pollinations to get the desired hybrids. Okay, pollen pistil interaction to get the desired hybrids, it can be asked in exam. So pollen germination by the dusting some pollen from the flowers such as pea, chickpea, rotularia, balsam, vinca on a slideshow. You can see a drop of sugar solution about 10%. About 50 to 30 minutes you will see there is the pollen tube formation from the pollen grains. A breeder, okay, now a breeder is interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties. Artificial hybridization is one of the approaches of crop improvement program. So artificial hybridization is one of the approaches to improve the crop improvement program. In such process experiments, it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from contamination from unwanted pollen. This is achieved by the emasculation and bagging technique. Okay, so emasculation and bagging technique, why does it take place? Because to prevent from unwanted pollen. Okay, if the female parent bears bisexual flowers, removal of anther from the floral bud before the anther dehiscence, anther, that is releases the pollen grains that matures. Okay, release of pollen grains using a pair of forceps is necessary. This step is known as emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. This process is known as bagging. So in artificial there is emasculation, then there is bagging. When the stigma of the bag flower attains receptivity, mature pollen grains collected from anthers of the male parent and are dusted on the stigma. So bagging, then there is a dusting and the flowers and rebag. So in artificial there is emasculation, bagging, then dusting and then rebagging, and the fruits allowed to develop. If the female plant produces unisexual flowers, there is no need for emasculation. If the female plant is unisexual flowers, produces unisexual flowers. The female flower buds are bagged before the flowers open. Female flower buds are bagged before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen, and then the flower is rebagged. Okay. So this is it. Now the next thing is the double fertilization. Now double fertilization, it involves the syngamy and triple fusion. Okay, after entering one of the synergies, the pollen tip releases the two male gametes into the cytoplasm of the synergies. Okay, into the cytoplasm, into the one of the synergies. So one of the male gametes moves towards the egg cell and fuses with its nucleus, then thus completing the syngamy. This results in the formation of diploid cell, the zygote. Okay, the other male gametes moves towards the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus, pen. Triploid primary endosperm nucleus, that is pen. As this involves the fusion of three haploid nuclei, is termed as triple fusion. Since two types of fusion, syngamy and triple fusion, takes place in an embryo sacs, where does the two fusion takes place in the embryo sacs? The phenomena is termed as double fertilization. An event this is unique to flowering plants. The central cell after triple fusion becomes the primary endosperm cell, that is spec. The central cell after triple fusion becomes the primary endosperm cell. So the central cell after triple fusion, the central cell after triple fusion becomes the primary endosperm cell, that is PEC, and develops into the endosperm, while the zygote develops into the embryo. So primary endosperm develops into the primary endosperm cell develops into the endosperm, while the zygote develops into an embryo. Okay. So now you can see fertilized embryo sac showing zygote and primary endosperm nucleus, that is PAN. So you can see degenerating synergies are shown, zygote, that is 2N, primary endosperm cell, that is PEC peg. Then after that there is primary endosperm nucleus, that is 3N, that is pen. Okay, and on downward side there are degenerating antipodal cells. So you can see the shape of the today's embryo. Okay, the stages in embryo development in dicot, that is shown in reduced size as compared to the adult one, as compared to the A. Okay, now so fertilized embryo sac showing zygote and primary endosperm nucleus, that is pen, and stages in embryo development in the inner dicot, right? Now the post fertilization, that is structure and events. So following double fertilization, events of endosperm and embryo development, maturation of ovules into seeds and ovary into fruit are collectively termed as the post fertilization events. So the endosperm development precedes embryo development. So endosperm development precedes embryo development, right? Okay, so endosperm development precedes embryo development, right? Why? Because the primary endosperm cell divides repeatedly and form a triploid endosperm tissue. These are the food reserves, right? The cells of this tissue are filled with reserve food materials and are used for the nutrition of the developing embryo. Now in most common type of endosperm development, that is PEN, primary endosperm nucleus undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called the free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. So subsequently the cell wall formation occurs and the endosperm becomes cellular. Okay, the number of free nuclei formed before cellularization varies greatly. The coconut water from the tender coconut that you are familiar with is nothing but free nuclear endosperm. Okay, so coconut water is just the free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm, right? Endosperm may be either be completely consumed by the developing embryo, that is pre-groundnut and beans before seed maturation, or it may persist in the mature seeds, that is castor and coconut. castor and coconut and be used up during seed germination. So split open seeds of the castor, peas, beans, ground fruits of coconut and look for the endosperm. In each case, find out whether the endosperm is persistent in cereals, that is wheat, rice and maize. So embryo. Now we are going to study the embryo. So in embryo develops at the micropyle end of the embryo sac where the zygote is situated. Okay, so zygote is situated at the micropyle end, right? Okay, so most zygotes divide only after a certain amount of endosperm is formed because it gets nutrition from the endosperm. Because it gets nutrition from the endosperm, right? This is an adaptation to provide assured nutrition to the developing embryo. Though the seeds differ greatly, the early stages of the embryo development embryogeny are similar to both monocot and dicot, right? Now this depicts the stages of embryogeny in dicot cotyledon. The zygote gives rise to the pro-embryo and subsequently the globular or embryo, GH, and then heart-shaped embryo and then mature embryo. So okay, so as you can know, so a typical dicot embryo consists of an embryonal axis and two cords. The portion of the embryonal axis above the level of cotyledon is epicotyl, which terminates in the plumule or the stem tip. The cylindrical portion below the level of cotyledon is hypocotyl, that terminates at its lower end in the radical or root tip. The root tip is covered with a root cap, right? The embryos of monocot possess only one cotyledon. So embryos of monocot possess only one cotyledon. In the grass family, the cotyledon is called scutellum, that is situated towards the one side that is lateral of the embryonal axis. At its lower end, the embryonal axis has the radical and root cap enclosed in an undifferentiated sheath called the and it is known as the undifferentiated sheath, not differentiated sheath, called the coleorhiza. The portion of the embryonal axis above the level of attachment of scutellum is the epicotyl. The epicotyl has a shoot apex and a fluid primordia enclosed in a hollow foliar structure, the coleoptile. Now okay, so this is it, and we are going to study this diagram right now. So a typical dicot embryo, as you can see, there is a plumule, that is shoot tip, and above the level of the cotyledon is the hypocotyl. Below the level of the cotyledon is the hypocotyl. Above the level of the cotyledon is the epicotyl, and then radical, and then there is a root cap, and then we are looking at the longitudinal section of an embryo of the grass. Okay, the first one was the typical dicot embryo. Now second is, you can see the scutellum, the one sided one cotyledon, then coleoptile, right? Then shoot apex, and then epiblast, then radical, then root cap, and then coleorhiza. So coleorhiza or coleoptile, these are the undifferentiated sheath, right? Then radical, root cap, and then coleorhiza, right? So this is it. Now we are going to study the seed. So what about the seed? So what about the seed? So in angiosperms, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Okay, seeds are formed inside the fruits. A seed typically consists of a seed coat, cotyledons, and an embryonal axis. So, okay, the cotyledons of the embryo are simple structure, generally thick and swollen due to the storage of food reserves as in legumes. Okay, now the cotyledons of the embryo are simple structures, generally thick and swollen. So cotyledons are thick and generally they are swollen due to the storage of food reserves as in legumes. So mature seeds may be non-albuminous and ex-albuminous. That is, non-albuminous seeds have no residual endosperm as it is completely conceived during the embryo development. Example, pea and groundnut. Okay, now albuminous seeds retain a part of endosperm as it is not completely used up. So it is not completely so what it say, like, so it is not completely So where we at, like, okay. So mature seeds may be non-albuminous except albuminous. So non-albuminous seeds have no residential endosperm as it is completely consumed during embryo development. So groundnut and pea. Now albuminous seeds retain a part of albuminous seeds retain a part of endosperm as it is not completely used up during embryo development. Example: wheat, maize, barley, and castor. So albuminous seeds retain a part of endosperm as it is not completely used up during embryo development. So wheat, maize, barley, and castor. Occasionally, in some seeds such as black pepper and beet, remnants of nucellus are also persistent. This residual persistent nucellus is the perisperm. Okay, I repeat it again. Occasionally, in some seeds such as black pepper and beet. So black pepper and beet, remnants of nucellus are also present. That is, this residual persistent nucellus is the perisperm, right? So seed integuments of ovules harden as tough protective seed coats. Example: the micropore remains as a small pore in the seed coat, right? This facilitates entry of oxygen and water into the seed uh, water into the seed water into the seed during germination. Okay, as the seed matures, its water content is reduced and the seeds become relatively dry, that is 10 to 50% moisture by mass. Okay, the genetic metabolic activity of the okay, so where we add, like, the genetical metabolically. Okay, so as the seed matures, its water content is reduced and seeds becomes relatively dry, that is 10 to 50% moisture by weight. The general metabolic activity of the embryo slows down. It slows down the embryo. So the general metabolic activity of embryo slows down. The embryo may they may enter a state of inactivity called the dormancy, or if favorable conditions are available, adequate moisture, oxygen, and suitable temperature, they germinate. Now, and as ovules mature into seeds, the ovary develops into fruit, right? The ovary develops into a fruit. That is the transformation of the ovules into seeds and ovary into fruits proceeds simultaneously. Okay, the wall of the ovary develops into the wall of fruit called the pericarp. So wall of the ovary develops into a wall. So wall of the fruits, these are the wall of the ovary that develops into a pericarp. The fruits may be fleshy as in guava, orange, or mango, or maybe dry groundnut and mustard. So many fruits have evolved mechanism for dispersal of seeds. Recall the classification have evolved have recall the classification of fruits and their dispersal mechanisms between number of ovules in an ovary and the number of seeds present in a fruit. Okay, so now in most plants, by the time the fruits develop from the ovary and other floral parts degenerate and fall off. However, in a few species such as plants, strawberry and cashew, the thalamus also contributes to fruit formation. Such fruits are called false fruits, and most fruits however develop only from the ovary, and these are called the true fruits. Although in most of the species fruits are the results of fertilization. Although in most of the species the fruits are the results of fertilization, there are few species in which fruits develop without fertilization. Also sees a better adaptive strategies for dispersal of new habitat. Okay, so now, okay, so where we were at, also the seeds have a better adaptive strategy for discovery to the new habits and help the species to colonize in other areas as they have sufficient food reserves. As they have sufficient food reserves, the young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to the young embryo being so being okay the so being products of sexual reproduction, they generate new genetic so they generate new metabolic conversion data okay combination leading to variation. Seed is the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for so dehydration and dormancy of mature food. Okay, so where we are at. So how long? So how long do the seeds remain alive after they are dispersed? This period again varies greatly in a few species. The seeds within a few months. Seeds of a large number of species live for several years. Some seeds can remain alive for hundreds of years. And there are several orders of very old yet viable seeds. The oldest is that of Lupinus arcticus excited from Arctic tundra. The seed germinated and flowers. The flowers after an estimated record of 10,000 years of dormancy. A recent record of 2,000 years old viable seed is one of the date palm and Phoenix dactylifera discovered during the archaeological excavation at King's Harris Place near the Dead Sea. So Phoenix dactylifera discovered. Phoenix dactylifera discovered during the archaeo. Okay. So after completing a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following questions, like how many eggs are present in an embryo sac, how many embryo sacs are present in an ovule, uh, how many ovules are present in an ovary, how many ovules are present in a typical flower, how many flowers are present on a tree, and so on. Okay, so some plants in which the fruits contain very large number of seeds. Orchid fruits are the examples, right? Each fruit contains thousands of tiny seeds. So orchid fruits. Similarly, in case in fruits of some parasitic species such as Orobanchia and Striga, have you seen a tiny seed of ficus? Now, how large is the tree of ficus developed from that tiny seed? How many billions of seeds does each ficus tree produce? Can you imagine any other example in which such a tiny structure can produce a large biomass over the years? Right. Now the apomixis and polyembryony. So the apomixis and polyembryony. So although seeds in general are the products of fertilization, a few flowering plants such as some species like Atracia and grasses have evolved a special mechanism to produce seeds without fertilization. To produce seeds without the fertilization, right? Called apomixis. What is what what is food production without fertilization called? So what is a food product? Okay. Although seeds in general are the products of fertilization, a few flowering plants such as some of the alternate have evolved mechanism to produce seeds without fertilization. Their apomixis. What is food production without fertilization called? Thus, apomixis is a form of asexual reproduction that mimics the sexual reproduction. Okay, there are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction division and develops into the embryo without fertilization, more often as in so in some in some species in some species the diploid egg cell is formed without reduction division. The diploid egg cell is formed without reduction division and develops into the embryo without fertilization. So more often as in citrus and mango. You can see varieties of the nuclear cells surrounding the embryo sac start dividing. Some of the nuclear cells surrounding the embryo sac starts dividing, protrude into the embryo sac and develop into the embryos. In such species, each ooid contains many embryos. Occurrence of more than one embryo in a seed is termed as polyembryony. Take out some seeds of orange and squeeze them. So this is the polyembryony. Now hybrid varieties of several of our food and vegetables crops are being extensively used, right? Cultivation of hybrids has been tremendously increased productivity, and one of the problems of hybrid is that the hybrid seeds have to be produced every year. If the seeds collected from the hybrids are sown, the plants in the progeny will segregate and do not maintain hybrid characters, and so do not maintain the hybrid characters. Production of hybrid seeds is costly, and hence the tobacco and hence the cost of the hybrid seeds become if these hybrids are made into so production of hybrid seeds is costly, and hence the cost of the hybrid seeds become too expensive for the farmers. Okay, if these hybrids are made into apomictics if these hybrids are made into apomictics, there is no segregation of characters. So there is no segregation of so the, so there is no segregation of and do not maintain hybrid characters. Okay, the production of hybrid seeds is costly, and hence the cost of the hybrid seeds become too expensive for the farmers. If these hybrids are made into apomictics, there is no segregation of characters in the hybrid progeny. After a year he does not keep on the using the hybrid seeds to raise new crop. Year after year he does not have to buy hybrid seeds every year. Because of the importance of apomixis in hybrid seed industry, research active research is going on in many laboratory around the world to understand the genetics of the apomixis and to transfer apomictic genes into a monohybrid varieties, right? So this was your all the chapter is being done. Let me know if anything is left. I will cover it.